everybody. Today we are going to look at Neisseria gonorrhoeae. Okay. First of all, we have an introduction to Neisseria genus. Okay. This Neisseria genus consists of gram-negative, aerobic, non-sporulating, non-motile, oxidase positive cocci that are arranged in pairs. Hence, they are known as diplococci. Okay. And this Neisseria genus consists of pathogenic as well as non-pathogenic species. Pathogens are Neisseria meningitidis and Neisseria gonorrhoeae. These two are strict human pathogens, and this Neisseria meningitidis causes meningitis in humans, and gonorrhoeae causes gonorrhea, that is a sexually transmitted disease. And the Neisseria meningitidis is also known as meningococcus, and Neisseria gonorrhoeae also known as gonococci. Okay. And non-pathogenic species are Neisseria lactamica and Neisseria subflava, which are normal floras of human mouth and respiratory tract. Okay. Next, we have identical or unique features of Neisseria gonorrhoeae. Okay. Here, Neisseria gonorrhoeae causes venereal disease known as gonorrhea. Venereal means sexually transmitting disease. Okay. So, Neisseria gonorrhoeae causes sexually transmitting disease known as gonorrhea, and this gonococci is first described in gonorrheal pus by the scientist Neisser in 1879. Okay. Then next, its morphology. This Neisseria gonorrhoeae are gram-negative and appear as diplococcus with adjacent sides concave, being typically kidney-shaped. Here you can see gram-stained picture of Neisseria gonorrhoeae. Then this Neisseria gonorrhoeae are non-sporing and non-motile, and these organisms possess pili on their surface. Pili means it is a hair-like structures with several micrometer long, which facilitate the adhesion or attachment of organism to the mucosal surface of the host. And this pili also promote virulence by inhibiting phagocytosis. So, this Neisseria gonorrhoeae are gram-negative, non-sporing, non-motile, and have pili on their surface, and they are typically arranged in pairs, hence known as diplococci. And you can see the gram-stained picture of Neisseria gonorrhoeae. Okay, next is about cultural characteristics of gonococci. Okay, here Neisseria gonorrhoeae are aerobic but may grow aerobically also, and their optimum temperature being 35 to 36 degrees Celsius and pH is 7.2 to 7.6. And this range of temperature as well as pH is required for their maximum growth and multiplication. Okay, and it is necessary to provide 5 to 10 percentage carbon dioxide for their improved growth. Then enriched medias like chocolate agar and mullen hindern agar are used for the cultivation of Neisseria gonorrhoeae. When we inoculate this organism into these enriched media, which produces small, round, translucent, convex, slightly ammonate colonies with a finely granular surface and lobate margin. Okay. These are the general or common colony morphology of Neisseria gonorrhoeae. Okay, and according to Neisseria gonorrhoeae, 4 types of colonies have been recognized. They are named as T1, T2, T3, and T4. Among them, T1 and T2 form brown colored colonies, small brown colored colonies. Okay, and the strains that form T1 and T2 type colonies are pileated and auto-agglutinable as well as virulent. And T3 and T4 type colonies are large, granular, non-pigmented. And the strains that form T3 and T4 are non-pileated, form smooth suspension, and are avirulent, that is non-pathogenic. Okay. So, the cocci that form T1 and T2 type colonies are pileated and virulent, and the cocci that form T3 and T4 type colonies are non-pileated and non-pathogenic, right? And the T1 and T2 types are also known as P plus as well as P plus plus, and the T3 and T4 are P minus because they are non-pileated. Okay. And then for selective isolation of Neisseria gonorrhoeae, thiomartin media is used, which contain vancomycin, colistin, and nystatin. These are antibiotics that improve the selectivity. Okay. That is, the antibiotics like vancomycin, colistin, and nystatin present in the selective media which inhibit the contaminants or prevent the organism other than Neisseria gonorrhoeae. Okay. Next is about their biochemical reactions. Here, Neisseria gonorrhoeae are catalase and oxidase positive. That is, they can produce catalase as well as oxidase enzymes. And H2S production test is negative with the Neisseria gonorrhoeae, and this Neisseria gonorrhoeae does not reduce nitrate to nitrites. Okay. Then this Neisseria gonorrhoeae can ferment only glucose with the production of acid, not gas. These are the some biochemical reactions which are shown by Neisseria gonorrhoeae, right? Okay, next we have antigenic properties. Here, gonococci are antigenically heterogeneous, and this gonococci have antigenic structures like pili, outer membrane protein, then an endotoxin, and also produce an enzyme known as IgA1 protease. These are the antigenic structures that present in the Neisseria gonorrhoeae. Okay. And about pili, as already discussed that the pili are hair-like structures having several micrometer long and which act as a virulence factor also by promoting attachment to the host cell as well as inhibiting phagocytosis. Okay. So pili is an antigenic structure as well as virulent factor. They act as virulent factor by inhibiting phagocytosis as well as facilitating attachment of cocci to the host cells. Okay. And this pili are composed of repeating peptide subunits. These peptide subunits are known as pilins. It is a protein pilins. Pilins are the subunits of pili. Okay. And next is about outer membrane protein. As gonococci consists of three type of outer membrane protein as antigenic structure. They are named as protein 1, protein 2 and protein 3. Among them, protein 1 and 3 act as a ligand or receptors for attaching coccus to the host cell and also form transmembrane channels known as porins for their exchange of molecules across the outer membrane. Okay, so type 1 and 3 are act as the receptors for attachment to the host cell as well as they act as a channel for the exchange of molecules across the outer membrane. Okay. Then type 2, that is protein 2, is related to opacity of gonococcal colony. So it is known as opacity associated outer membrane protein. That is, the strain with the opacity associated protein form opaque colony and those lacking it form transparent colony. Okay, as protein 2 containing strains form opaque colony and the strains do not have opacity associated protein, they form transparent colonies. Okay, and this opacity associated protein, that is type 2 protein, also responsible for the attachment of cocci to the host cell. Hence these outer membranes present in the gonococci are also act as a virulence factor, thereby attachment helping in the attachment to the host cell. Okay. Then another antigenic structure is endotoxin, that is present in the cell membrane or cell wall of the gonococci, which is responsible for the toxicity of gonococcal infection. It is lipo-oligosaccharide in nature, mainly present in the cell wall and also act as a virulent factor by forming toxic effect during gonococcal infection. Okay. Then another one is IgA1 protease enzyme that is produced by gonococcus which inactivate IgA antibody. Okay. So these four types of antigenic structures are present in the gonococcus, and they are also act as a virulent factor by helping in pathogenicity in gonococcal infection. Okay, next is about their resistance. Here, gonococcus is a very delicate or unstable organism, so they can easily or readily killed by heat, drying, or any other antiseptics. And it is a strict parasite and dies in 1 to 2 hours in exudates outside the body. That is, when the gonococci released from their host into their environment, they die within 1 to 2 hours because they are strict parasite. They can't live without a host. Okay, then in cultures, the cocci die in 3 to 4 days but survive in slant cultures at 35 degrees Celsius if kept under sterile paraffin oil. That is, in cultures they survive only for 3 to 4 days, and in slant cultures they can survive several days at 35 degrees Celsius if they are kept under sterile paraffin oil. Okay, then these cultures of gonococci may be preserved for many years if they frozen immediately or stored under minus 70 degrees Celsius. And this gonococci contains several types of plasmids like cryptic plasmids, and this cryptic plasmids containing genes that code for beta lactamase enzyme that cause this gonococci resistant to penicillin. That is, some of the strains of gonococci are resistant to penicillin due to the production of beta lactamase, and this production of beta lactamase enzyme is encoded by the plasmid present in the gonococci. Okay, so these are about resistance. Okay, next we have pathogenicity. Here, Neisseria gonorrhoeae is a causative agent of venereal disease known as gonorrhea. Venereal means sexually transmitting disease. Okay, and this gonorrhea that has been known since ancient time. That is the oldest infection, and the disease is acquired by sexual contact. That is, the organism enter into a host by sexual contact, and the first step in the infection is the adhesion of this gonococci to the urethra or other mucosal surface. Okay, and the pili that are present in the gonococci are help or involved in their adhesion to the host cell surface. Okay, then after adhesion they penetrate through the intracellular spaces and reach the subepithelial connective tissue by the third day of infection. Okay, and after the incubation period of 2 to 8 days, in men the disease starts with the acute urethritis with the mucopurulent discharge containing cocci in large number. In females also, the gonococci adhere or attach to the urethra as well as cervix uteri and causes urethritis. But vaginal mucosa is not usually affected because of acidic pH of secretion present in the vagina. Okay. And the urethritis present in the females is extended towards Bartholin glands, endometrium as well as fallopian tubes, and cause infection in that areas also. And in females, the infection with the gonococcus also lead to pelvic inflammatory diseases and salpingitis. Salpingitis means it is the inflammation of fallopian tube. Okay, so this salpingitis as well as pelvic inflammatory diseases lead to sterility. And in some persons, peritonitis may develop with the perihepatic inflammation. Peritonitis means is the inflammation of peritoneum with the perihepatic, that is liver cell inflammation, which is known as Fitzhugh-Curtis syndrome. And here some of the women are asymptomatic, that is they carry the gonococci without any symptoms. Hence they are considered as a reservoir of gonococci. Okay. And besides these infections, this gonococci also causes proctitis, conjunctivitis, blood invasion, ophthalmia neonatorum. Okay, the proctitis means it is the inflammation of rectum. Conjunctivitis, it is the inflammation of conjunctiva. Then gonococci also invade the bloodstream and causes some skin lesions like Okay, next is about epidemiology. Here, gonorrhea is an exclusively human disease. It is only confined to human beings and there being no natural infection in animals. Okay. So, the only source of infection is a human carrier and less often a patient. And the existence of asymptomatic carriage in women makes them a reservoir serving to transmit infection among their male contacts. Okay, that is, the females are the carrier of gonococci and act as a reservoir, and they transmit this infection to their male contacts. Okay, and the mode of transmission is almost through venereal or sexual contact, and only one non-venereal infection is that of Thalmia neonatora. Okay, then fomites do not play any significant role in their transmission because the cocci die rapidly outside the human body, that is, they are strict parasites. That's all about epidemiology. Okay, next is laboratory diagnosis. It is necessary to collect appropriate specimen for the diagnosis. Here, urethral discharge or urethral swab or endocervical swab can be collected as a specimen. And if no urethral discharge is available, deposits of urine, that is centrifugal deposits of urine, can be collected for the diagnosis of gonococci. Okay. And the methods of diagnosis include microscopy, cultivation or culture, serology, as well as molecular methods. And on microscopic method, gram staining can be performed, which shows gram-negative diplococci. That is, if the specimen contain gonococci, when we perform gram staining, which shows gram-negative diplococci. But this gram staining procedures is unreliable in women, as some of the genital normal flora have the same morphology as gonococci. So it is not reliable for the human for the diagnosis of gonococcal infection. Okay, and the use of fluorescent antibody microscopic method become more specific and sensitive for the diagnosis of gonococcal infection. Fluorescent antibody technique means the use of fluorescent coating antibodies against this antigen present in the gonococci. Okay, so microscopic method is not a routine diagnostic method for gonococcal infection. So, culturing is an important method for the diagnosis of gonococcal infection, and for culture, specimen should be inoculated immediately on collection. If this is not possible, specimen should be collected with the charcoal impregnated swab and sent to the laboratory in STARS transport media. Okay, this is because we know that this gonococci are strict parasite. They are die within 1 to 2 hours outside the body, right? So the specimen should be inoculated as soon as possible. And for the cultivation, the enriched media like chocolate agar, mullen Indian agar, and selective media like Thayer Martin agar can be used for the isolation of gonococci from the specimen. And the growth is identified by. Okay, next is about their treatment. In ancient time, that is when penicillin was introduced, all the strains of gonococci were highly sensitive to penicillin. That is, their minimum inhibitory concentration was 0.005 unit per ml. Okay, but from 1957, this sensitivity or susceptibility towards penicillin become decreased, and their minimum concentration or minimum inhibitory concentration become 0.1 unit per ml. Okay, and after that, that is from 1976, the gonococci produces beta lactamase or penicillinase enzyme and to become highly resistant towards penicillin, and these penicillinase producing Neisseria gonorrhea have spread widely. At that condition, we have used ceftriaxone and ciprofloxacin as a drug of choice for the treatment of gonococcal infection. And at that time, some strains also become resistant towards ciprofloxacin. So, instead of the ciprofloxacin, we use azithromycin or doxycycline for the treatment. Okay, so now azithromycin or doxycycline with the ceftriaxone are the drug of choice used for the treatment of gonococcal infection. Okay, next is prophylaxis, that is how to control the gonococcal infection. Okay, we know that this gonorrhea is a venereal disease, right? So the measures we have to take to control the gonorrheal infection mainly include general measures such as early detection of cases, contact tracing, health education, etc. Right? And a non-venereal disease known as ophthalmia neonatorum, which is controlled by a method known as Craig's method. Craig's method is the practice of instilling or adding 1% of silver nitrate solution into the eyes of all newborn babies so as to control the non-venereal disease known as ophthalmia neonatorum. Okay, then this gonococcal infection does not confer any clinical immunity towards the or in the host body. So secondary infection with the same organism is possible, right? And here vaccination has no place in the prophylaxis. Only general measures are taken to control the gonococcal infection, avoiding the avoiding the sexual contact with the infected ones, or contact tracing or early detection of cases. Health education are the measures we have to taken to control the gonococcal infection. Okay. So that's all about Neisseria gonorrhoeae that causes a non-venereal infection known as gonorrhea. Okay, thank you.